Hello, guys. Good morning, everyone. I hope all of you are doing fine. Good morning, Vishal. Good morning. Good morning, Keethana. Good morning, Mahima. Good morning, Siva. Good morning. Good morning, Tarun. Good morning, everyone. So, how are all of you doing? Hi, Mukesh. Okay, you're from Ahmedabad, City of India. Me too. You will give me a chance to speak with you. Okay. Yes. Yes, whatever your queries are, Mukesh, you can give it on your chats. No, I'll read it and I'll answer you. Okay. Right. So so today we'll be talking about the GPCR signaling pathway. Okay. So a lot of question comes in CSI net exam and in gate also related to the GPCR signaling. So GPCR signaling is very vast and there is so much to cover in it. So in this particular session only, I'll cover all the GPCR related concepts and how the signaling takes place and what are different pathways for which the GPCR is responsible for. Okay. So there will be free sample papers also for this particular unit. So you can download it from the link given in the description, right? So the topics for the discussion today would be from Unit 2, that is GPCR signaling. So where we'll be studying about the molecular mechanisms through which the signal transduction majorly takes place. Generally, the signal transduction, how it takes place, that molecular mechanism we'll be studying about. Then we'll be knowing about what is G protein, to which family it belongs, and what are its components. Then is the G protein signaling activation and termination. After that, we'll be speaking about the classification of the trimeric G protein and their effectors. Um, then we'll be covering the prototype, which is majorly being studied a lot regarding GPCR, that is the beta adrenergic pathway. Then the hormone induced activation and inhibition of adenylyl cyclase. After that, we'll be studying about the cholera toxin, that is what is the mechanism of cholera toxin and how it is responsible, like how GPCR is connected to cholera toxin, right? Then coming to the GPCR activating phospholipase C. Then GPCR signaling, which is involved in nitric oxide or NOCGMP signaling, where basically dilation of the arteries takes place. And then we'll be studying about the sensory transduction in vision. So how our retina basically works and how we are able to visualize things. And through GPR signaling, we'll be studying that. So yesterday we had discussion on Unit 3. So here is a question, like yesterday we left at this question. So I hope all of you would have solved this question. So I would be expecting answers from all of you. Yes. Yesterday's answer, okay, Mahima says answer is 1 and 5. Okay. Anybody else? Make us learn about the varieties of questions in the CSI and how should we approach them. Yes, of course, Einstein. I'll let you know like what all question comes from this particular unit from GPCR. Okay. In stepwise manner only we'll study GPCR so that you can understand it in a better way. So answer also Mahima explanation also she gave. She said that because poly A polymerase is a template independent enzyme. Yeah, right, Mahima, you are correct. So Keethana says answer is B. Soumya says answer is C. Okay. So most of you says that the answer is C. So yeah, all of you are correct. Answer is C only. So yesterday, like we have studied about the stepwise procedure which takes place during polyadenylation of the 3' end of the pre-mRNA. So poly A tail. Wait a second. Let me change the color. Yes. So polyadenylation and the poly-A polymerase, which is basically responsible for addition of the adenine, polyadenine tail in the 3' end of the pre-mRNA. It is a template independent enzyme. It doesn't require any template for addition of the adenosine at the 3' end. Okay, so first option is correct. Then option second, it says the poly-A polymerase catalyzes the addition of AMP from DATP to the 3' end of the mRNA. This is wrong statement, because poly-A polymerase it catalyzes the addition of AMP from ATP. It's not DATP, okay, at the 3' end of the mRNA. So speaking that, we can see that the option 5 is the correct option. So wherever we can see that there is only one option where the option 1 is given. So option 1 is correct and option 5 is correct. So regarding this question, the correct option would be option C. I hope all of you got clear with this, right? Also, it uses ATP as a substrate for binding. Yes, Mahima, correct. So this was a very easy question, I guess, right, for all of you. So then coming to the first topic of this particular session regarding GPCR. So we will be studying about the molecular mechanism signal transduction. So what is the general mechanism through which any signal transduction takes place, right? So for the signal transduction, there are 6 major things which are required. Like first is the specificity should be there. So whichever signal transduction is there, it is highly specific to its ligand, right? A particular specific type of ligand only binds on the basis of the complementarity. That can only bind to a particular receptor, and no other signal can fit inside the receptor, and the signal transduction won't take place. First is the specificity. Second comes the amplification. So enzyme activates enzyme. So here you can see from the figure it's given here. So this is the figure. So here you can see that there is a signal coming. So it activates first enzyme. Then this particular enzyme, produces further signals and activates enzyme 2. And many of the amplified version of enzyme 2 is getting signal, and there are many enzyme 2 which is getting activated. After this activation of enzyme 2, enzyme 3 gets activated. So in a cascade manner, right, multiple enzymes are getting activated through just one signaling. So amplification of signaling takes place during any signal transduction, right? Third is the modularity. So in modularity, what happens is, so there is a signal that basically gives signal to a particular receptor. This receptor has a amino acid which gets phosphorylated. So this receptor gets phosphorylated. In turn, this receptor phosphorylates the other receptor, and this receptor gets phosphorylated. And after the phosphorylation of this receptor, which it activates the other receptor, and this receptor also gets phosphorylated. So in the form of phosphorylation, the receptors are getting activated, and the ultimate response is being produced here. So in modularity, it is being said that the proteins with multivalent affinities form diverse signaling complexes from the interchangeable part. So there will be many parts for these receptors. So it can phosphorylate at multiple places, right? And the response is ultimately being generated like this. Then comes the desensitization and adaptation. This is very important, because whichever signal transduction takes place, wherever the signal is taking place, that signal needs to be inhibited also. So here comes the role of feedback inhibition. So receptor activation triggers a feedback circuit. When any signal is not being required by the body, so on the basis of the requirement of the organism or the cells, what happens is there is a deactivation of particular signal also takes place, and this basically happens through desensitization, right? So it basically shuts down the receptor or removes it from the cell surface. So basically this desensitization in GPCR majorly happens through receptor mediated endocytosis. So here is a here is a GPCR receptor. Okay. It is present. This is the plasma membrane. So this GPCR receptor, when it binds to a ligand, then at that time the signal transduction in the intracellular signal transduction, it takes place. Apart from that, then only the endocytosis process also is taking place. Like simultaneously, endocytosis process is also taking place. So when the signal is has performed its activity, all the activity is being performed. After that, immediately this particular receptor, it gets endocytosed. Like it is being engulfed by the cell itself, and it comes inside the cell. Right after the signal is completed. So desensitization plays a role here. After that comes the integration. So in integration, you can see in the figure. So the figure says there are two signals, right? So integration basic definition is that the ability of a system to receive multiple signals and produce a unified response. So even if a system gets multiple signals, it will produce one effect only, one response only would be produced. So here we can see that when the GPCR signaling is taking place, then RTK signaling doesn't take place, right? So RTK doesn't take place when GPCR signaling is taking place. And when RTK is taking place, then it inhibits GPCR signaling, and GPCR signaling is not taking place. So one signaling at a time takes place, and there is a crosstalk between multiple signaling so that one response is produced at a time, right? Then coming to, yes, so there are multiple names with which GPCR is being called. So in exam, like you can be given that a serpentine receptor or a G protein coupled receptor, GPCR, or 7 transmembrane segment, 7TM receptor. So all these names are given to GPCR, right? Coming to the 3 essential components of GPCR. So first is the receptor. So receptor is present extracellularly, like it is present outside the plasma membrane. So it has 7 transmembrane helical segment in it. So 7 times it like crosses the plasma membrane, right? Then comes the effector. So effector is the another component of GPCR, where the this effector is basically present in the plasma membrane. It is an enzyme that basically generates an intracellular secondary messenger. So in case of GPCR, there will be multiple secondary messenger you will come across. The predominant one is the cyclic AMP, right? The CAMP that we call. Then is the third component of the G protein coupled receptor is the G protein itself. So G protein is majorly present intracellular, like within the cell. So this is basically a trimeric guanosine nucleotide binding protein. So it is a trimeric, it is present in its trimeric form because it is having 3 subunits, right? So this is yeah. So this would be the G protein. So the G protein would be there. So there would be 3 subunits of this G protein. There would be alpha, which would be present, and there would be beta subunit, and there will be gamma subunit, right? That will be beta and gamma. So it is present in its trimeric form within the cell. So, and yeah, and this is linked to the receptor. So this particular trimeric protein is linked to the receptor, that is GPCR. So in here you will come to know about how many 3 types of G proteins are there. That is G stimulatory is there, G Q is there, G transduction is there, and G inhibitory is there, GI, right? GI. So 1, 2, 3, 4 type of G proteins would be there that you will be coming across in whole GPCR signal, right? What are their different effects? What are their different roles you will be knowing? Then here it is given that there is a receptor class pathway. So this particular table shows that the G protein coupled receptor, the ligands with which this particular G protein coupled receptor binds are, it could be epinephrine, it could be glucagon, it could be serotonin, vasopressin, ACTH, that is corticotropic hormone is there, and adenosine, and many others in case of mammals takes place, right? Then comes the odorant molecules, light, mating factors, etc. So in humans, there are basically like human basically encodes for 1,000 members of this family of receptor, GPCR family of receptor. It is majorly being specialized for transducing messages diverse as like light that we like through light that we visualize. In that also GPCR is responsible. Signal from the GPCR is responsible. For the smell also GPCR is responsible. For taste also GPCR is responsible. And there are multiple signal transduction that GPCR it performs, and multiple hormones to which it binds, right? So the receptor here is the 7 transmembrane alpha helices. So this receptor is majorly present like outside the plasma membrane. It is an extracellular part. Then the cytosolic domain, it is basically associated with the membrane that is tethered with the trimeric G protein. So within the cytoplasm, the G protein is present. So here is the GPCR, right? This is the plasma membrane. This is the receptor part. This part is the receptor which is present in extracellular portion. And then here in the cytoplasm, there would be the G protein, right? In the cytoplasm, there will be G protein. And G protein would be present, it would be attached to the G protein in the trimeric form, right? So this is how the GPCR basically looks like, the whole structure. Then is the signal transduction. So it is involved in multiple signal transduction processes where it also produces the second messenger. So in second messenger pathway, it is involved where CAMP is being produced. There are multiple secondary messengers that are being produced, like such as cyclic AMP, such as IP3, such as DAG is there, right? And linked ion channels and MAP kinase pathway. So in all these pathways, in all these signal transduction, GPCR is involved. So this is the basic overview of GPCR. So here comes the characteristic of G proteins. So first, this is the revision tool, that is the Conceptica wheel, through which you can see for yourself like what are the different characteristics of G protein. So first is the G protein binds to guanine nucleotide, either GTP or GDP. So when the G protein is bound to GDP, then it is present in its inactive form. And when it is bound to GTP, it is present in its active form, right? Then comes the second definition. They are heterotrimeric, consists of these 3 different polypeptide subunits, alpha, beta, and gamma. So I've told you earlier, it is a trimeric protein consisting of 3 particular subunits, alpha, beta, and gamma, right? Then the other definition, it says that the heterotrimeric G protein are held at the plasma membrane by the lipid chains that are covalently attached to the alpha and gamma subunits, right? So the G protein, which is held at the plasma membrane, it is attached to the lipid chain. The plasma membrane is attached, and then it is also attached to the other subunits, that is alpha and gamma, through the covalent bonds, right? Then it says that the guanine nucleotide binding site is present on the G alpha subunit. So the GDP or the GTP with which the G protein is attached, that part is basically present in G alpha. So GDP or GTP basically binds to G alpha subunit of G protein, right? Then it says that the GTP bound conformation, the G alpha subunit has a low affinity for G beta gamma. So trimeric protein, it is a trimeric protein consisting of alpha, beta, and gamma subunits. Now, when the GTP binds to G alpha subunit of G protein, then what happens? This alpha subunit, it gets dissociated from the beta and gamma subunits. So it is the alpha subunit which is dissociating, not the beta gamma one, right? And then it says that each dissociated subunit is free to activate an effector protein and produce a secondary messenger. So when this G alpha it is attached to GTP, then it gets activated and it will further give signal to an effector molecule. There will be different types of effector molecules to which these different G proteins would be binding. And when this effector molecule gets activated, it produces a secondary messenger. Secondary messengers could be of multiple types. It depends on which effector is getting activated, right? It could be CAMP, or it could be the IP3 DAG. All these things, you will be coming across that when you will be studying about different types of GPCR signaling, right? Then comes the classification is based on the G alpha subunit and the effectors to which they couple. So the classification of G protein coupled receptor, it is basically based on the G alpha subunit. Which type of G alpha subunit is present? It could be G I alpha, G inhibitory alpha, G S alpha, that is G stimulatory alpha. It could be GQ alpha, or it could be G transducin alpha, right? So all these different types of G proteins are present, G alpha subunits are present. On that basis, the GPCR is being classified. And other way of classification of GPCR is the effectors. So there are multiple effectors that present in case of GPCR signaling. So on the basis of the GS or the G alpha protein, there are how many? There are 4 types of G proteins. So first it is given here. So here is GS alpha. This is the stimulatory one, right? So this basically activates the effector molecule adenyl cyclase. So adenyl cyclase effector molecule is being stimulated by this particular GS alpha stimulatory protein, right? Then GQ family. So GQ alpha. What does this do? This basically activates the effector molecule PLC beta, right? This also we'll be studying later on. Then comes the GI inhibitory one, GI subunit. So this basically functions and it inhibits the adenyl cyclase. So the effector molecule at which it acts and it inhibits it, it is basically the adenyl cyclase, right? So in this way, there are multiple G protein. Yes, there are multiple G proteins present on which basis the GPCR is being categorized into different types. So here it is giving the general way in which the GPCR, basically the signal transduction in GPCR takes place. So the heading says that the G protein signaling activation and termination. So this figure basically tells how the GPCR signaling gets activated and how it gets terminated, right? So major example we are taking for is epinephrine. So epinephrine is a hormone, right? It binds to the receptor. So this is a GPCR receptor. So amino terminal is present at the extracellular part, and the carboxyl terminal is present in the cytosol. So here at the amino terminal, basically this ligand will bind, and the G proteins are basically attached to the receptor or linked to the receptor at the carboxyl terminal, right? So this is being given. This is the G protein, which is present in its trimeric form. So this is the GS alpha, and this is the beta and the gamma subunit, right? So when the ligand it binds to the receptor, then what happens? There is a conformational change which takes place intracellularly. So the G protein, the conformational change majorly takes place in the G protein. So before the activation, what happens is GS alpha is already bound to GDP. Now when the ligand comes and the signal transduction takes place, this GS alpha, the GDP gets dissociated and GTP bounds to the GS alpha subunit of the G protein. And what happens, this GS alpha when gets activated by binding of GTP, then what happens, this GS alpha, it dissociates from the beta and the gamma subunit, right? So GS alpha gets dissociated, and this dissociated GS alpha activated form, then it what does it do? It binds with the adenyl cyclase. So this is the effector molecule. So binding to adenyl cyclase, it activates this particular effector molecule. And what happens is this adenyl cyclase, it converts ATP into CAMP, that is cyclic AMP, right? This cyclic AMP is a secondary messenger which has multiple functions. So this CAMP, it activates the PKA, protein kinase A. So here is the structure of the CAMP which is being given. So what is given in the structure is that how from ATP CAMP is majorly being formed. So this is the ATP molecule. You can see there are 3 phosphates with which the ribose sugar is attached, and there is adenine here. Then what happens? The PPI, that is the phosphate group, is being removed. So 2 of the phosphate groups is being removed, and 1 of the phosphate group is remaining, which in the cyclic form it attaches to the 3' end, right? So the major definition of the CAMP is basically it is an adenosine 3' 5' cyclic monophosphate. So there is single phosphate present, and it is present in the cyclic form and attached to 5' end and the 3' end. So this is the basic structure of cyclic AMP, right? So coming to this step, when the cyclic AMP is being produced, it activates the protein kinase A. This protein kinase A, it does the phosphorylation of various cellular proteins, right, which causes the cellular responses due to the epinephrine, right? What kind of responses? Such as epinephrine is basically a fight and flight hormone. So when you're in such kind of condition, like where you're panicking and all that stuff, at that time, epinephrine is the hormone which conducts the GPCR signaling, and all those responses takes place, such as somebody is after you and you have to run. At that time, the energy is being provided by this particular GPCR signaling, and epinephrine hormone is responsible basically for that, right? Then coming to once the CMP is being produced, the concentration of CMP is increased in the cytosol. So for the GPCR signaling to get inhibited, this cyclic AMP is also required to be its concentration has also to be maintained. So lowering its concentration, there comes another enzyme, that is the cyclic nucleotide phosphodiesterase, like PDE we call it. So this enzyme, what does it do? It converts the cyclic AMP into 5' AMP. That is the normal adenosine monophosphate, right? This majorly takes place, and the concentration of CMP in the cytosol, it decreases, and at that time the CMP is degraded, reversing the activation of pKa. So when the activation of pKa is not required, at that time the concentration of CMP need to be lowered in the cytoplasm. For that, this particular enzyme is responsible, right? I hope you're understanding what I'm saying, right? Everything is clear in this figure, right? This particular slide. You can give me a thumbs up if you're clear, if you want me to repeat also. Okay, it's clear. Okay, Anup says it's clear. Devanshi says it's clear, right? So we'll move further. So here comes the classification of the trimeric G protein and their effectors. So this table basically tells what is different classification of the trimeric G protein and the effectors which are involved. So first is the GS alpha, that is the G stimulatory alpha protein, G protein. So this basically responds to the receptor that it responds to is the beta 1 and the beta 2. So let me tell you the receptor, the GPCR receptor is basically of beta 1 type, beta 2 type, alpha 1 and alpha 2 type. So these are the 4 forms of this receptor which is basically present inside our body. So this G protein, G stimulatory alpha, is it basically responds to the beta 1 and beta 2 form. GI alpha, it basically responds to the alpha 1 form, and GQ alpha, it basically responds to the alpha 2 form, right? Then comes the effector molecules to which these G proteins basically acts upon. So associated effectors is for GS alpha, it's adenylyl cyclase. For inhibitory one, it's the adenylyl cyclase, and there is like another type of effector also, K plus channels is also present, potassium channels. For this, basically G beta and gamma activates this effector, right? And comes the olfactory one. For this, the adenylyl cyclase is the associated effector. For GQ alpha, it's the phospholipase C, and for GT, that is transducin alpha. For that, the CGMP phosphodiesterase, PDE basically, is the associated effector, right? So the second messenger that is being produced due to the activation of effector molecule by these G proteins are For GS stimulatory G protein, where adenyl cyclase is the effector molecule, when it gets activated, it produces the secondary messenger CAMP. So most of these GPCRs, the secondary messenger is basically CAMP. So in case of stimulatory, the concentration of CAMP is basically being increased. For inhibitory one, the concentration of CAMP need to be decreased, right? And for olfactory one, also the CAMP need to be increased. While coming to this one, the phospholipase C, the GPCR in the phospholipase C signaling, for that the secondary messengers are not like cyclic AMP. They are basically IP3 and DAG-driven signal transduction, right? For transducin, the CGMP is the major secondary messenger which is being produced. For this, like here, the concentration of CGMP is basically being decreased, right? And the examples of receptors is being given here. So this table is very important for you. You can note it down, or you can also like refer to your standard book. Leringer is the book from which I have Lodish is the book, yeah, from which I have taken this particular table. And this table is very much important, giving a lot of information about the characteristics or the classification of the GPCR, right? Yes. So it is again a reminder for all of you that Shaurya batch, it is being starting from 4th of March, and it would be targeting the CSIR NET 2022, the upcoming 2 exams in this particular year. So here will be many facilities that would be provided to you, such as there will be RAID classes, interactive online classes would be there, and doubt solving would be there. Then hard copy would be provided to you, study materials would be provided, and video backups would be there. So if you want to know more about this batch, and so for that you can call us on the toll-free number that is given on your screen, and you can also email us, right? So coming to the basic pathway in GPCR, it is a prototype of GPCR signaling, which we call as beta adrenergic pathway, right? So it is same as like how GPCR takes place, but it is given here brief, like which effector molecule will get activated, and which kind of G protein is responsible, and how it affects, and how secondary messenger is being produced, and what all effects it will have. So firstly, the epinephrine binds to its receptor, right? So this is the receptor. This is the adrenergic receptor. So adrenergic receptors are basically of 4 different types, like alpha 1, alpha 2, beta 1, and beta 2. So this adrenergic receptor, it basically depends upon which like G protein will get activated. On that basis, the hormone will bind. So if epinephrine binds to its receptor, it would be specific. It could be alpha 1, alpha 2, or any, right? Then here comes the in the cytosol, there is present the G protein. So G alpha S is already bound to GDP here. So you have been shown here that it is already bound to GDP. So when the signal takes place, and when the hormone gets bind to the receptor, this GDP is being dissociated, and GTP gets bound to GS alpha. So GS alpha gets activated, and it gets dissociated from the beta and gamma subunit. After that, it binds to the adenyl cyclase, which is the effector molecule. This adenyl cyclase gets activated, and it converts ATP into CMP. So concentration of CMP is being increased in the cytosol. So this CMP activates PK, and PK is responsible for the phosphorylation of various cellular enzymes responsible for cellular responses which takes place due to epinephrine, right? So coming to the hormone-induced activation and inhibition of adenyl cyclase, right? So here it is being given, the stimulatory hormones are being given to you. That is, stimulatory hormones are majorly like epinephrine for GPCR. It could be epinephrine, it could be glucagon, it could be ACTH. ACTH is basically adrenocorticotropic hormone, right? It is majorly being produced in the anterior pituitary. And the inhibitory hormone is being given, that is PGE, that is the prostaglandin E, which is majorly produced in adipose tissue. So this is the inhibitory hormone, and adenosine. So these 2 hormones, like these different types of hormones, they have different effect on or different response they produce inside our cell. So first is that when the stimulatory hormone it attaches to the receptor, it binds to the receptor. The receptor for the stimulatory hormone it is given here, and the receptor for the inhibitory hormone is being given. So there will be different receptors for different kind of hormones, right? So activation takes place here, and the GDP bound GS alpha, the GDP gets dissociated, GTP gets bound, and all these activities takes place, and it bounds to the adenyl cyclase. Now, in stimulatory response, what happens? The adenyl cyclase is activated. While in case of inhibitory response, what happens is the GDP it remains bound to the G alpha part, and it is not being converted into GTP, and as a result the adenyl cyclase is not being activated and further response doesn't take place. So in this way the inhibition also takes place simultaneously and the stimulation also takes place, right? So this particular figure basically tells how the binding of different types of hormones and it leads to the inhibition and stimulation of GPCR, right? So differences basically comes the difference in effects on the tissues when the receptor is bound by different hormones. It basically depends on three particular factors: the type of receptor it is binding to, the hormone which type of receptor it is binding to. Second is the type of G protein is getting activated. Like if it's GS, then stimulation which will take place. If it's GI, then inhibition will take place, right? And third is the set of the pKa, that is the phosphokinase A, which particular type of pKa molecule it's being targeting, right? The enzyme to which it is targeting. Mahima says that CGMP reduction triggers a nerve impulse indicating that the light has been detected. Yes, it comes in the rhodopsin one. Yeah, Mahima, we'll be discussing that also, right? So here comes, yes, the cholera toxin mechanism of action. So it basically works on this inhibition kind of response, right? In cholera toxin, what happens is GPCR signaling is being inhibited. So how does it happen? So studying about the cholera toxin, getting an overview what the cholera toxin is about, because in exam related to cholera toxin also comes, like what are different subunits of cholera toxin. So here in the concept wheel, the basic general characteristics of cholera toxin has been given. So first it says that the toxin produced by the bacterium Vibrio cholerae. So cholera toxin is basically produced by the bacteria Vibrio cholerae. Everybody knows that, right? So the cholera toxin is an oligomeric complex. It's an oligomeric complex consisting of two subunits. Like it would have A subunit and B subunit. So B subunit it has 5 parts. Like there are 5 B subunits which are present, and there is single A subunit which is present. So it is basically being denoted as like AB5, right? Secondly, it says that the subunit B, it binds while the subunit A activates the G protein. So subunit B binds to the receptor, and A part, the subunit A, for that it basically provides a gateway, the B part, because the B part is being binding to the receptor. So it will provide a gateway for the A to pass through. And the A is the one which will bind to the G protein, and it will activate the G protein. Conformational changes will take place in G protein, and it will further activate the adenyl cyclase, right? Then it says that the toxin modifies the G alpha S protein. So the toxin which is getting inside the A part, the A subunit, it will do? It will modify the G alpha S protein. We'll know like how does it modify in the next slide. By the ADP ribosylation. This ADP ribosylation is very important. How the G protein gets modified by the toxin, it's by ADP ribosylation in the intestinal epithelial cells, right? The result of which is the alpha remains, the G alpha, it remains in the active state. So the result of which is the G alpha subunit, GS alpha subunit, it will always remain in its active form. That is, the GTP will be bound to it, and the adenyl cyclase will be active all the time, and there will be like CAMP would be producing inside the cytosol. So the concentration of CMP will be increased in the cytosol. As a result, it will lead to the opening of the channel, that CFTR is the channel basically, through which what happens, the chloride ions, it moves out, and it will provide the nutrition to the bacteria, right? Yes. So electrolytes and like everything, water and everything, it will be removed from the cytosol and it will be provided as a nutrition to the bacteria, and we'll be feeling dehydrated. The person which is infected will feel dehydrated because of this. So here in the figure, it is saying that, yes. So it says that, as we know that the cholera toxin, it has two subunits, A and B. So this B subunit basically binds to the GPCR receptor, and it provides a passage for A subunit. This A subunit, what it does, it does the conformational change or modifies the G alpha subunit. As a result, the GTP, when it binds to the G alpha subunit, and GTP gets dissociated. Now what happens, the G alpha, when the toxin is there, the G alpha, the GTP is not being removed from the G alpha. It will be binding to G alpha continuously. As a result, it will activate the adenyl cyclase. So adenyl cyclase will be present in its activated form always. As a result, the concentration of the CAMP inside the cytosol, it will increase, and this, what it will do, it will open up the CTFR channel. As a result, the chloride ions, it is being gets out, and the sodium ion channel also, it's open, and sodium and chloride is being provided as a nutrition to the bacteria, and the person which is infected gets dehydrated, and water is being also removed from the body. So all these symptoms the person will be going through. So what do you understand by GDP ribosylation? So this modification in the G alpha subunit is basically due to the GTP ribosylation. So how ADP's ribosylation takes place, that we'll be studying. So here you can see in the figure that this is the phosphate group is there, adenine is there. So this is the structure of NAD+, right? The bacterial toxins causing cholera, it catalyzes the transfer of ADP ribose moiety of NAD to an arginine residue of GS, okay, stimulatory G protein. So what does it do? This particular, yes. So this basically transfers, this is the arginine part, this is the arginine residue which is present in the G S alpha. So with this arginine residue, this particular NAD molecule, it will get bind to it. As a result, the GS alpha, it will always be in its activated form, right? GTP would be bound in it. This is majorly being done by the cholera toxin. So cholera toxin, what does it do? It removes this nicotine. This nicotine is being removed by the cholera toxin, and only this part, this particular part, this is being attached to the arginine residue of the GS alpha, right? As a result, the GS alpha, there is modification takes place in the GS alpha, and it's always in its activated form. GTP would be attached to it. As a result, it will bind to the adenyl cyclase. Adenyl cyclase will also be in its activated form always, and the concentration of the cyclic AMP, it will increase inside the cytosol, and then the sodium ion channels and the chloride ion channels and the water will be removed from the body, and it will be provided as a nutrition to the virus, to the bacteria. So this is what happens in case of Vibrio cholerae infection, right? This is the general mechanism. Mima says, so basically ADP ribosylates GS alpha GTP can activate adenylate cyclase, but incapable of hydrolysis. It's bound GTP, yes, Mahima, and hence the adenyl cyclase remain locked in its active state. Yes, Mahima, you're correct. That's what I was explaining. So coming to the next part, that is the GPCR activating phospholipase C. So GPCR also activates another effector molecule, that is the phospholipase C molecule, right? Till now we were studying about the adenyl cyclase, and now we have come to the effector molecule. The effector molecule has changed, that is the phospholipase C, and here you will see that the G protein has also changed. So here the major G protein which gets activated is basically the GQ, right? And the secondary messengers here produced are also different. The secondary messengers here produced are basically the DAG, diacyl glycerol, and the other one is the IP3, right? So how this mechanism basically works and what all effects it have, we'll be studying it through this particular diagram. And this diagram is also given in your lecture. You can go there after I have explained it and study for yourself, right? So what happens is the hormones, it binds to the receptor, right? This is the hormone, it will binding to the specific receptor here. Now what happens, the GDP bound GQ protein, this basically gets activated. The GDP gets dissociated here and the GTP is being bound, right? Then what happens, this activated GQ, it binds to the effector molecule phospholipase C, right? After getting attached to the phospholipase C, this phospholipase C effector molecule gets activated. What does this phospholipase C do? It cleaves the phosphatidyl inositol 4,5 bisphosphate to inositol triphosphate, right? So bisphosphate has been converted to IP3 and a DAG molecule is being released. That is the diacyl glycerol. So two types of secondary messengers are being produced. One is the DAG and another is the IP3, right? What does these secondary messengers do? So what happens, this IP3 which is being released, it binds to a specific receptor on the endoplasmic reticulum. Binding to the endoplasmic reticulum, what happens is there is the release of the calcium ions. So inside the endoplasmic reticulum, the calcium ions are present. A lot of calcium ions are present inside the endoplasmic reticulum. So inside the endoplasmic reticulum, the concentration of calcium ion is very high. So when this IP3, it gets attached to the receptor on the endoplasmic reticulum, these calcium ions, the channel opens and the calcium ions comes out of the endoplasmic reticulum into the cytosol, where the concentration of calcium ion is low. So calcium ion is also one of the kind of secondary messenger, because this also activates various enzymes inside our body, right? So when this calcium ions comes out to the cytoplasm, then this calcium ion binds to the DAG molecule, right? So, sorry, it binds to the protein kinase C. So DAG molecule is like when the PLC, that is the phospholipase C, it cleaved the phosphatidyl inositol 4,5 bisphosphate into two secondary messengers, that is the IP3 and the DAG. The IP3 one, it bound to the receptor on the endoplasmic reticulum and led to the open of the calcium channel, and the calcium ions from the endoplasmic reticulum comes to the cytoplasm. Another that secondary messenger, that is the DAG, it remains attached to the plasma membrane, right? This is the DAG molecule which remains attached to the plasma membrane. Now when the calcium ions comes to the cytoplasm, it will bind to the phospholipase C, right? Phospholipase C protein it will bind to, and DAG will also bind to this phospholipase C. So together both of these DAG and calcium ions, it activates the protein kinase C, right? Protein kinase C is activated by both of these, calcium ion and the DAG protein, and at the surface of the plasma membrane it happens. Then what happens, this phosphorylation of the cellular proteins by the protein kinase C takes place. It produces some of the cellular responses, or the hormones are being produced. So various kind of cellular responses are being produced when the phosphorylation takes place of the protein kinase C, and this further phosphorylates various enzymes in our body, and as a result various cellular responses takes place, right? So this is the general structure of the DAG protein, and this is the general structure of the IP3 secondary messenger, right? So here it is written that the DAG, it basically activates the protein kinase C, PKC, and the IP3 it basically opens the calcium ion channels in the endoplasmic reticulum. So these are the two major functions, right? Yes. Other one is the nitric oxide, that is NOCGMP signaling pathway. So here also GPCR signaling is required, right? So you can see in the diagram here what is it has been given, that there is a lumen of blood vessel, right? So blood vessel is there, right? The outer layer of the blood vessel is basically made up of endothelial cells. This endothelial cells, it basically acts as a barrier between the lumen of the blood vessel, like inside the blood vessel, and the tissues which are present outside. So how can I explain that? This is the blood vessel, right? So outer layer of this blood vessel, it is basically consists of endothelial cells, right? And here comes the lumen. This is the lumen. So I'm giving you the general picture how this particular pathway is basically taking place inside the body. So this is the blood vessel. Inside is the lumen. The outer layer of this blood vessel is basically consists of the endothelial cells, and there would be tissues present here, right? Tissues would be present here. So here majorly taking the example of smooth muscle cells, right? Cells would be present. So this is how this particular signaling will take place. I'm giving you the idea of how the location is there. So what happens here is, so acetylcholine is the ligand here, the particular signal molecule. So it binds to the acetylcholine GPCR, G protein coupled receptor, a particular kind of receptor for this type of ligand. Then what happens? This after binding of acetylcholine to this GPCR, it leads to the activation of the phospholipase C, the thing that we studied, the effector molecule phospholipase C that we studied earlier. So phospholipase C plays a part here also, right? So phospholipase C effector molecule, it gets activated. Then what does it do? It'll form two types of secondary messengers. So here the role of IP3 basically comes. IP3 secondary messenger comes here, and it will bind to the calcium calmodulin protein. So this is another kind of acidic protein. So calmodulin is another kind of acidic protein with 4 high affinity calcium ion binding sites. So this is highly its affinity is very much high for the calcium ions. So whenever there is a sudden change in the concentration of calcium ion inside the cell, then what happens? This calmodulin protein, it binds to the calcium ions, right? So it detects the change in the concentration of the calcium ion inside the cell. It regulates the activity of calcium dependent enzymes. So whosoever enzyme is its activity is being regulated through calcium enzyme, this calmodulin molecule, it binds to the calcium ion and activates, or it basically stimulates the activity of those calcium dependent enzymes, right? So then when this calmodulin protein, it gets activated, what does it do? It affects Yes, it produces the NO signaling. So it acts upon the NO synthase. This is an enzyme, right, which is basically responsible for converting arginine to citrulline. As a result, the NO, that is nitrogen oxide, is being produced inside the body. So what happens is, the events of the NO signaling is downstream target of this calcium calmodulin is the NO synthase. So this NO synthase enzyme is the one to which this calmodulin protein, it basically stimulates its activity. As a result, this NO synthase, it synthesizes the NO gas from arginine. Then what happens? This NO gas, it basically, this is the NO gas it produced. So this NO gas, it basically binds to the NO receptor. NO receptor is majorly present in the smooth muscle cell, right? So inside the smooth muscle cell, there would be NO receptor, to which this particular NO gas that is being produced, it will bind to it. As a result, what happens is the smooth muscles, that it basically causes the relaxation of the muscle cells, the smooth muscle cells, increasing the concentration of CGMP. So what happens is GTP molecule, it is being converted into CGMP when this NO gas, it basically binds to the NO receptor. When the concentration of CGMP, it increases inside the smooth muscle cell, then it activates the protein kinase G, is another effector molecule, right? This activation, it will lead to the relaxation of the muscle cells, right? So in the arteries, arteries of the heart tissue, it gets dilated by this process only, and this signaling only, and it can supply the blood to the tissues. So whenever we require a lot amount of blood inside the body to the tissues, so at that particular time, this particular pathway will take place, and the dilation of the blood vessels will take place, right? So coming to the another pathway which is being stimulated by the GPCR. This is the sensory transduction in vision. Like how are we able to visualize different kinds of things in their colored form and which wavelength. So all this thing is also controlled by the GPCR signaling. So let me give you a brief introduction here right now. So what happens in humans, the retina that is there in the humans, it is of two types. It consists of two types of photoreceptors are present there, which are primary recipient of the visual stimulation, right? So two types of cells or the photoreceptors which are present in human retina are the rod cells and the cone cells. So rod cells, it basically stimulates, it is stimulated by the weak light. Like it doesn't require enough of light, like even in the moonlight also, at the night time also, like it gets stimulated, right? Then it is basically being used at the night time. So at the night time, the rod cells present in our retina, it gets activated, right? And it lacks the color vision. So at the night time, we basically see all the things in the gray scale, like there is no color there. So the another type of cell which is present in the retina is the cone cell. So this cone cell, it is being stimulated at the high intensity of light. Whenever the high intensity of light is present, then only these cone cells get stimulated, and it is majorly involved in the color vision, right? So so signals on these two photoreceptors, these are basically being processed as a part by the part of the brain which is known as the visual cortex. So inside our brain, there is a part known as the visual cortex. So all those responses or the signals which is being produced by these photoreceptors, the signals, it is directly being gone to the visual cortex, and then we are able to visualize different kind of things, right? So the major proteins which are involved in this is basically a GTP coupled signal transduction. So here the transducin G protein will act, right? So the major proteins which is involved in the GTP bound GPCR signaling is, first one is the rhodopsin would be there, right? So for rhodopsin, it's written that this is a G protein coupled receptor, right, which is present in the rod cells. So it is present in basically in the rod cells and are activated by light. So when light falls on this rhodopsin, which is a G protein coupled receptor, this is the major receptor which basically lies on the extracellular part. This is what it's saying. This is the rhodopsin, right? And here would be the GT, G alpha T would be present here, and it will be bound to the beta and gamma part. So this is the receptor it is talking about. So the receptor here is the rhodopsin, which is majorly present in the rod cell and is activated by light. Then it consists of the light absorbing pigment. This rhodopsin also consists of a light absorbing pigment, that is 11-cis retinal. So and this 11-cis retinal is basically covalently bound to the 7 spanning protein, that is opsin. So this is the 7 spanning protein here, and this is here it is saying that this is opsin. So you can resemble with the structure of the GPCR, the what part it is basically talking about. So opsin is the 7 spanning protein to which this pigment, this is basically a chromophore pigment, due to which we can see, at the time of light, we can actually see the things in the colored form. So this 11-cis protein, which is the pigment present inside the rhodopsin, it is a light absorbing pigment. This is covalently bound to the 7 spanning protein, that is opsin, right? Then comes another protein which plays a major role here, is the transducin. This is a trimeric G protein coupled to rhodopsin. So inside the cytosol, this trimeric G protein will be present, and it will be coupled to the rhodopsin receptor, and it is often called the transducin GT, as I've told, and it is found in the rod cell. This is also found in the rod cells. Then coming to the CGMP phosphodiesterase, that is PDE. So this is another this is another enzyme that converts the cyclic GMP into 5' GMP, right? So normal form of GMP it will convert when the activity, we want the activity to get inhibited at that particular point of time, this PDE would be playing its role, right? So here is the general structure of the rhodopsin is being given here, which consists of the 11 cis retinal moiety. It is a pigment chromophore, and this is the structure of the opsin. Yeah, it is covalently linked to the opsin, and when this gets activated in the presence of light, so light induced isomerization takes place, and this particular 11 cis retinol gets converted into the meta rhodopsin 2. So rhodopsin gets converted into meta rhodopsin 2, and here the opsin protein, it gets activated, right? So here is a simple step it is taking place. So here comes the rhodopsin receptor, right? It is bound to the opsin protein. So when the and it also consists of the 11 cis retinal part, that is the chromophore. So when the light is being falling here on the rhodopsin receptor, then what happens? The photons, they basically acts as a ligand here. So what happens? This 11 cis retinal, it gets activated and gets converted into 11 trans retinal. Like isomerization takes place in cis form, it gets converted into the trans form, right? When this transform is being formed, it activates the rhodopsin and converts it into meta rhodopsin 2, which is the activated opsin protein, right? When this gets activated, it further activates the GT transducin, right? So the transducin trimeric protein, it activates it. As a result, when this transducin molecule, it gets activated, what does it do? It activates the cGMP phosphodiesterase PDE enzyme, right? When this PDE enzyme gets activated, what is its role? It basically converts the cGMP, the cyclic GMP present inside the cell, into normal GMP, that is 5' GMP. As a result, what happens is the hyperpolarization due to the sodium ion channel closure takes place. So before, when the concentration of cGMP inside the cytosol was very high, at that particular point of time, the sodium channels were open. But when the this particular phosphodiesterase PDE enzyme, it gets activated, what does it do? It converts the cGMP into 5' GMP. As a result, the concentration of the cGMP inside the cytosol, it reduces. As a result, the sodium channel, which was open earlier, it will get closed, and the hyperpolarization of the cell will take place. So inside the cell, the voltage would get converted into minus 71 millivolt, right? So hyperpolarization takes place inside, it will become more negative, because the channel is closed and there is no sodium ion which is coming inside. So inside the cell, the hyperpolarization takes place. And this due to this hyperpolarization, what happens? It decreases the release of neurotransmitters. So these neurotransmitters are majorly responsible when like if there is so much of light present, we don't need that much of signaling, so that the neurotransmitters are not required at that time, because we are able to visualize things properly. But at the dark time, when there is no light, at that particular point of time, it is required that the neurotransmitters, like its concentration increases, and we are able to see things properly. Because at the point, when the light is very much high, at that particular point of time, we don't need neurotransmitters. So there is a decrement of neurotransmitters which takes place due to this particular pathway, right? Okay, so this much, is everyone clear with it? So then only we'll move further, or you want me to repeat it? Is everyone clear with this particular pathway of rhodopsin which is taking place? Okay, Dilshad said it's clear. Devanshi, it's clear. Okay, so now we'll be moving further. So here in the form of a diagram also, I have explained how this process basically takes place. So stepwise procedure, I told you already in the presence of light, how the rhodopsin basically works and how the signal is basically being produced. inside the body. Now here is the diagram. So here in the diagram you can see this is basically the light which is being falling on this particular rhodopsin receptor. So inside the rhodopsin receptor, there would be the opsin protein would be present, right? And this rhodopsin would be attached to the opsin protein. So this opsin protein, it basically absorbs the light and gets activated. When this opsin protein it gets activated, then what happens is it binds to the inactive GDP bound GT protein mediating exchange of the GDP with GTP. So what happens is the trimeric protein with which this particular receptor is attached, this is the rhodopsin, right? This is rhodopsin, which consists of the opsin protein. In the presence of light, it gets activated. When it's get activated, then what happens is the trimeric protein with which this receptor is bound to, the trimeric protein here is basically the transduction G protein, right? GT alpha. So for that, what happens is the GDP here with which it is already attached, when the activation takes place, the GDP gets dissociated and it will bind to GTP, right? So now this trimeric protein is basically bound to GTP, right? When it is bound to GTP, this is basically GT alpha, then it will activate the inactive PDE. So the PDE in inactive form, it is basically consisting of 4 subunits. Like there will be 2 gamma subunits and alpha and beta subunits. So alpha and the beta subunits are the one which is required, and 2 gamma subunits, they are basically inactivating PDE, right? So what happens is when this GTP bound GT alpha, it binds to this inactive PDE, these 2 gamma subunits, it gets dissociated, right? This will get dissociated, right? It's written here, the third point. The the GT and GTP complex form dissociates inhibitory gamma subunits, and then what happens, it activates the CGMP phosphodiesterase PDE, right? So this inactive form, it will get converted into the active form of PDE. When this PDE gets activated, then what does it do? It converts the CGMP present in the cytosol into normal GMP. That is the 5' GMP it will convert. So what happens inside the cytosol, the concentration of CGMP, it will get lowered down, right? So when the concentration of CGMP is being lowered, it will lead to the closure of the CGMP gated ion channel. So this is this channel, the sodium potassium, the sodium calcium channel, it is there. So this ion gated channel, the CGMP was earlier attached to it, but when the concentration of the CGMP inside the cytosol, it is being lowered at that particular point of time, this gated ion channel, it will get closed. As a result, there will be no influx of the sodium or the calcium or any ion will take place. As a result, inside the cell, it would be the condition which would be created, it would be hyperpolarization condition, and it would be more negative, that is minus 71 millivolt would be the inside charge. Then coming to the dark adapted state. So when there is no light present, at that particular point of time, what happens is there is no light, okay? So this opsin protein, it will not get activated. As a result, the GT alpha, the trimeric protein, it will be attached to GDP. GTP won't be able to bind to it. It will be attached to just GDP. As a result, this PDE, it will remain in its inactive form. Since it is in its inactive form, this CGMP won't be converted into GMP, and the concentration of CGMP inside the cytosol, it would be very high, right? As a result, what happens, this particular CGMP gated ion channel, it will always be open, and the sodium and calcium ion, the influx will take place, and as a result, the depolarization takes place here. Right, and inside the charge would be more positive. It would be maybe plus 40 millivolt, maybe. Yeah. So this would be the charge inside. So it would be depolarization condition, and earlier it was the hyperpolarization condition when this opsin protein was activated. So this is the major rhodopsin pathway, which is basically being conducted by the GT alpha subunit. So I have explained you about the entire GPCR signaling, what are signaling it is basically involved in, and the classification on the basis of the different types of G proteins, and what all different types of signaling in which these G proteins are basically involved in. All these things I've explained. I hope this lecture is very much helpful for you all, and you have built up your concept now. And again, if you go to the standard books and you try reading all this, you'll have a better understanding regarding this, and you'll not miss out any of the question, whichever question comes regarding the GPCR in your test paper, right? So here are the question answers. So these are the two questions that I've picked up. Both of these questions have come in your CSIR NET paper. So this is a kind of homework for all of you. Now, tomorrow when I come at this particular time, I'll be coming up with the unit 5, that is developmental biology. I'll be discussing concepts regarding some certain concepts with which questions majorly come in CSIR NET paper. So at that particular point of time only, we'll be discussing these questions, right? So I hope all of you would have understood and will go through your books and try solving these questions, right? Okay, thank you very much. So there would be free sample papers also. So there would be feedback form. If you fill up that, after that only you can get your sample papers, right? So link is being given in the description, so you can go through it. Thank you very much.